Okay, so I've pulled together a few different things that we're up to. We can talk about any or all of them. Let me just say that I spend part of my time up here trying to interact with the cell, the stem cell people, the people over in Zilka, trying to get some bridges built here on the clinical side as well. So I'm hoping if anything resonates that people will ask me to expand on it, or if it's anti-resonating, that you tell me to get off of it. My day job is figuring out how to image live tissues, mostly developing embryos and applying that a little bit into clinical utility. We do angiography and just got Chinese FDA approval for our OCT vascular imaging for diabetic retinopathy. That's an image from that. The thing I was thinking I would tell you about today is not the retinal stuff, which I'm happy to expand on, and I've got some slides in there if you want to hear about it. But some of the work we've been doing trying to apply multispectral and lifetime imaging sort of to add dimensions to the sort of imaging we're normally doing, and also to tell you a little bit about the way we're trying to apply our multiplex labeling technologies that let us apply to both experimental animals and to clinical samples, multidimensional imaging of sort of spatial genomics sort of issues. So I'm going to draw on a group of sort of engineers meet biology people that are at USC. Some of them are more genetics, some of them are more mathematical or physicsy. I'm going to tell you a little bit about what the Al Mann Institute has been helping us develop. And I'll draw a little bit, I think, on the stuff that started here when Andrew Brown was in ophthalmology. So anyway, so my day job is trying to analyze the complex interactions that go about to make an embryonic axis, and then organogenesis and correct patterning, both of the tissues within the organs and then the interconnections, either vascular connections or neuronal connections. A lot of times, as we start to look at these things, instead of seeing everything happening in a very lockstep way, what we see is that there's tremendous variance. And the way that we've been trying to get around that problem with variance is to be able to study the same sample over time, and the same sample with multiple imaging modalities and looking at multiple labels, so that variance and what co-varies with it becomes the signal rather than the complication. And so I thought I'd tell you a little bit about the dead imaging and then a little bit about the live imaging. The dead imaging is our attempt to do spatial genomics and spatial proteomics by doing quantitative visualization of proteins and RNAs. And we've got tremendous ability now, even down to the single cell level, because I think this is some of the single cell data out of one of our colleagues' papers. But you've got cells across here, genes across this way, and the different things are log 2, or the log 2 6 up or 6 down in expression. That seems very quantitative. Of course, it's haunted by the statistics of how well they harvest the proteins or how well they harvest the message. And the problem is that most in situ technologies for looking at where those genes are expressed are inherently problematic. Okay, so all the tissues, every piece of biopsy, intravital looking in lung or kidney, whatever, there's always autofluorescence, right? And people get away from that in part by taking thin sections, but it's still there. So to get the signal from the in situ hybridization above the background, the typical approach is to use an antisense probe that hybridizes to the target RNA. The probe, instead of being a dye, is an enzyme. That enzyme processes some colorless molecule and makes it colorful. But the dose response of that is always nonlinear. If there's a little tiny bit of gene product, or you only develop it for a short time, there's no color. If If you develop it for too long, it saturates. There's a little bit of a linear range in between, but for the most part, it's not very linear. So this is a friend's paper. It's the same strain of the same mouse in the same gene at the same stage, and actually it's in the same paper. And I'm supposed to know that those two are the same pattern. So I would argue that this makes it hard to make a quantitative measure and to study covariance. And so a lot of our work starts from me complaining, and luckily when I was complaining in this case, it was in front of people that did DNA origami, try to fold things up and make smiley faces or other issues about out of pieces of DNA. This Niles Pierce and his colleagues at Caltech were working on hairpins, and we realized we could try to use some of their technology to make something that would give us a more linear gain. These hairpins actually are complementary to one another, so they'd like to bind like that. But instead of binding that way, they're sort of trapped in this metastable state. They're trapped there because all those base pairings are holding it together. And if they wanted to find one another, they'd have to open up all the base pairings and find one another in solution. And the chances of doing that before the right and left sides of the hairpin find each other is very, very low, because effectively these are at hundreds of molar and there's no way that the reagent is at that. And it stays that way until it sees an initiator, and that initiator is basically half of the sequence on one of these hairpins. So this A star binds to the A, it branch migrates up, so for every base pairing that's lost, it gains a base pairing. You end up with this, which opens up and leaves a half hairpin available, which is the initiator for that, and it explosively grows. It does it in a sequence-specific way. So here's 4 hairpin pairs, and the hairpin pairs in this case are labeled with different dyes. If we take all 4 of them and throw them in a tube and add blender extract of tissue, then they still run in this gel as these little hairpins. If I add the blue initiator, blue disappears from here and appears up there. Add the green initiator, it appears up here and disappears from there. So we've got the ability to amplify from a 1 mer up to a 200 mer in a sequence-specific way. And so we take advantage of that by putting these initiators onto the antisense probes. And now what you've got is a number of dyes on a leash. It's about a 200 mer that forms in this case. And what that does then is it gives you enough signal to see it, to see the gene product where it's localized. It doesn't matter how long you develop it, the hairpins diffuse into the tissue. So unlike my old life of trying to figure out which gene had to be developed for 32 minutes and which needed to be for 4 days, you throw the reagents in. In fact, this one spent 2 months in the refrigerator before it was photographed. So it does well. Long Kai up at Caltech has now gone up into the 200-plex using this by serially staining and restaining. And I think I may have some of his pictures coming up. But basically, you can get different genes to light up. The nice thing is that I've published a lot on the gene expression of some of these markers and where they're supposed to be. And if you look around, you could find cells that clearly didn't read my papers. So those cells aren't supposed to be there, and there's a whole bunch of other violations that are there. There's a few other approaches that are coming out now for doing these sorts of approaches. What we like about the hybridization chain reaction approach is that it gives us the amplification. The amplification's linear with the amount of gene product that's there to about 3 to 5%. The hairpins diffuse in well, so it works well on thick samples and on biopsies and things of that sort. And the multiplexing and all goes way up. The other approaches work, but we think lack something. And so we're still refining it and have new versions of this coming out all the time. So the good news is we can label it. The bad news is we label it with dyes, and the dyes always overlap one another. And so if we take a transgenic animal that we work on in the lab a lot, watching the vasculature form, if we put a GFP variant in the blood vessels and a YFP, a yellow fluorescent protein variant, being expressed in all the muscle cells, and you looked at it by eye, it would not be very striking, not as striking as a cartoon. The problem is that there's almost nowhere here where you could just get the GFP or just get the YFP signal. In fact, there's probably nowhere. So again, complaining at the Athenaeum at Caltech, somebody from JPL started laughing at me, and they said, well, all of our signals overlap. We do not, with a satellite, put a giant blue filter in front of the sun and irradiate the Earth with only one color and then take a picture. We have to take what comes back. And so they started sharing with me the ways that they collect an X, Y, and then the third dimension is wavelength. So an image cube that's a spectral cube. This lambda stack then can be interpreted to see what the combination of things are in each pixel. So we worked with them first to make a prototype. It ended up being a thing that developed into a instrument that Zeiss has been selling for the last decade or two. And basically it's taking the laser light that to excite the specimen, collects the fluorescence that comes back, and disperses it over a couple, over 32 channels. And so just to give you an example of the performances from the old prototype even, these are the individual spectral bands. In this case, I've labeled the specimen with a histone that goes to the nucleus, phalloidin, which stains the actin. And you can see that each spectral band has both of them there. The dyes overlap tremendously, but if you do this approach of linear unmixing, it can separate the two. There's two problems with that approach. One is that it involves an inversion of some matrices. It's time consuming computationally. And the other problem is that the noise is dominated by the worst channel. So if I wanted to image, say, inside of a person, and the signal I was trying to look at was a vanishingly small signal, that noise from that, just the statistics of that would start to haunt me. And you can see how even in this two-color thing, this is happening. The green gets separated from the red, but the noise that's here, because there's only a few photons per pixel in this, gets imposed into the red image. And so if I now started to separate out multiple fluorochromes, this would be a problem. Enrico Grattan and his colleagues came to our rescue, and two of his people have joined my lab, and so they've been instrumental in moving forward on this. What they realized is you can turn that inverse problem into a forward problem by taking the Fourier transform of the spectrum. So the green spectrum becomes that green dot in a Fourier plane. The red becomes the red dot. A 50-50 mix of the two becomes a dot halfway in between. So now graphical sort of arguments can hold. It works well with the normal scope and with others that we've been developing. And this just shows how trying to separate it out with glass doesn't work. Separating it out, the approach that we sold to Zeiss years ago, works pretty well, but takes a while, and has this noise problem. So there's no blood vessels here in the middle between those two vessels, but there's signal. And the hyperspectral phaser approach gives you dramatic speeding in the processing, because you don't have to invert the matrices, and you can see that the noise goes away. So what this lets us do now is to take multiple colors. In this case, it's a live animal that's expressing several different fluorescent proteins, a blue fluorescent protein, a yellow, a green, and then also, while we're doing it, we're collecting the autofluorescence, the signal from the tissue itself. This one, for example, is an autofluorescence. I think the next one might. Yeah, that's the autofluorescence in the gut area of this animal. That's related to its metabolism. And this is a structural fluorescence, not unlike the green fluorescence you get out of lung tissue and things of that sort. But you can see that we can look right through the autofluorescent retinal pigment epithelium of the eye and see the blood vessels inside. You can see the lumen and the soma of the vascular endothelial cells on the two sides. So that means we can start to see these things in autofluorescence. We could start to think about, or sorry, I should say, we can see things that are exogenous labels. Can we also see things that are there in autofluorescence? And as we start looking around, you can see signals like the autofluorescence of the collagen, things like NADH bound and free, folic acid, a whole bunch of things. If we look in the animal, it's always easier to get informed consent out of the zebrafish. So you can see the metabolic signals, you can see things like retinoic acid and retinol, folic acid, a number of different metabolic markers. So we've been starting to use this to endogenously see things like retinoic acid, which is used as a morphogen in the animal, but also used in a lot of different settings. And as we started looking into more and more hostile environments, though, I have to admit that the other lesson from Enrico Gratton is that lifetime imaging can be better than multispectral imaging. And so lifetime is the amount of time it takes for a fluorescent signal to give off its photon. So a chemist would think about a dye molecule in a ground state, it absorbs a photon, it gives off a photon, and it takes some nanoseconds for that to happen. That happens, for example, in different metabolic state of the tissue. That could change tenfold for some of the endogenous fluorochromes. So if we could measure that exponential and deduce what the lifetime is, we would be able to sort of read the physiology of the cells. So we use a pulsed laser, it's at 80 megahertz, so you could time the amount of time it takes each one of the photons to come out after it and build up that sort of histogram. That can be done, but it's relatively slow. What Enrico Gratton realized a number of years ago now is that this Fourier domain trick makes it a lot easier. So instead of having to look at curves and saying, oh, that's NADH when it's bound or when it's free, and you can see how dramatically different those are. But if you go through on each pixel and interpret which curve it is or where it is in between, what he realized is that bound and free NADH and FAG and a whole bunch of other things, once you take the Fourier transform, show up in different regions. And so you could really just read off on a pixel by pixel basis, based on where the colors mix, what the state is. So this is the approach we're now starting to use clinically, in fact, extending it a little bit further along. Just to give you an example, if I have a whole field of dye, like a field of view of a dye like coumarin, and I look at it on the Fraser plot, all those pixels would be at the same spot. So I could, in the software, put my pointer here and say, show me all the places that have that lifetime. Or I can point to something here and say, tell me what the lifetime is. This is the simplest case. If instead we go into a piece of retina, in that case, I think it is, then what you've got is something that's spread out in different regions. You can see that we could move around on this plot and see what's there. And well, I guess very few patients have GFP, although with lab workers, maybe that's no longer true. But you can see things like NADH bound and free, retinoic acid, collagen, retinol, so things that the ophthalmology department cares about. These are in response to an ultraviolet excitation. That would be a little bit of a problem because A, it's not that great to blind people with ultraviolet light in their eye, but also the ultraviolet gets absorbed by water. But we're lucky that that we can use ultra-fast lasers that put out the 80 megahertz pulse, and we use, instead of UV light, we use IR light to excite the dyes. Now that's IR, so it goes through the vitreous, so it'll go deep in tissue. You can get a nice focus at depth. You might be able to see below the surface and see a ulcer before it's an ulcer, or inflammation that could lead to something. So it's great. We have the laser. We use it all the time for our 2-photon imaging and our light sheet imaging. We could get a cheap SLO, and you combine the two, and well, no. Okay, so we have not yet done it. Craig Russell was not the first thing. I don't know if anybody's seen the movie, but I think, if I remember right, the eye patch changes sides during the movie as well.
Yeah.
So,
that's right.
So anyway, we're still getting the ANSI tests down and all. So, and so I feel like I have to point out, this is a joke, just so I don't end up getting in trouble. But spheroids are a way to get at retinal tissue without having to worry about the informed consent. And so over several months, these will laminate into something that'll make a retinal ganglion cell layer, photoreceptors, and so on. So what we've been doing in these spheroids and in other settings that I'll tell you about in a bit is exciting with the ultra-fast laser, collecting the fluorescence that comes back. The color of it doesn't tell you that much. It tells you a little bit. The intensity of it doesn't tell you much because the amount of intensity that the laser light that makes it in and back is sort of limiting. But if you can go after the lifetime, you can start to see in the Fraser plot, for example, the difference between NADH bound and free, so the oxphos versus glycolytic sort of metabolism. You can also follow a lot of other things that are in the tissue. So we've been using this to watch the metabolism both in spheroids and in intact experimental animals. We're just trying to get in place to where we can do it in human subjects. But if we look in at one of these retinal spheroids, then we can see where the photoreceptors are very glycolytic. We can see the other metabolism on a layer by layer basis. And we can watch then as the spheroid develops from just a month or two old to half a year old, and watch as it laminates out. So this is something that lends itself to ongoing evaluation of tissue metabolism and tissue state. So we've been going after a lot of these things. We want to understand the retinol metabolism. We want to understand the connective tissue. The order of that connective tissue can tell you a lot about what's happening to tissue integrity, and we can also see markers of inflammation and other things. So where we are right now is we have one person in the lab, Mimo, who is a Enrico Grattan alumnus, who thinks the entire world is lifetime. But he admits that as well as lifetime imaging, there's hyperspectral imaging for the RNAs, for example. There's a number of different modalities. So he's now willing to consider multimodal imaging, even though he knows the whole world centers on lifetime. His former lab mate is also in the lab, Francesco Cottrell. He believes the entire world is hyperspectral. And so we have negotiated a truce, and the truce is let's do both. So there's still some overlap in the spectra here in lifetime. There's some overlap here. So the idea is to try to do a sort of belts and suspenders sort of approach, using both of them, especially as we start to be signal starved because we're going into humans where we don't want to use too much laser light. This might be a way to be able to separate the signals. So instead of our commercial scope, we've been working on a sort of semi-home built scope. This a company out of Enrico Grattan's lab years ago has been making a much more open architecture sort of instruments. We've been modifying this. The idea is to couple this in to the accessory port into a bronchoscope or other endoscopes and be able to see things. So that's sort of our dream. We are getting pretty close. I think that the paperwork is almost there. This is looking at experimental animal data, cell by cell sort of resolution, looking at the lifetime and hyperspectral phaser. And by combining the spectral and the lifetime, you can see features like that's the collagen on the far side of the airway. that's invisible in the lifetime. So it's great, we can see that all the way through. You can see where the cells are, we can see some of the metabolism based on the hyperspectral, but here in the airway, it's really clear in lifetime. And what they've been now doing with this spectrofilm, so this compromised instrument, is starting to think about, after different exposures, nanoparticles versus organics versus other things, they can see regional differences in the airway and different responses in the airway. So what we're hoping to do now is to try to apply this to a number of different settings. We want to try to make a hyperspectral lifetime imager that might look below the surface and see the earliest antecedents of a foot ulcer or anything else. So what I've tried to sort of give you a little bit of a window is how I go from my day job to my sort of weekend job. That is, we really are trying to build up the tools that let us ask many, many questions of the same piece of tissue. We're trying to do that now in live cells and be able to see what the constituent parts, including metabolism and structural signals. And now with adding in the lifetime, we're hoping that we'll be able to sort of read the mind of the cells and see what the metabolic state might be. And I should just mention a lot of people involved in this. In the hyperspectral Fraser, it was Francesco, the Lifetime's Mimo, Vikas Trivedi played a huge role in it, and many of the others that are listed here played an important part too. So thanks for your attention.
Thank you. Go ahead. No,
Yeah, no, you first.
Well, I was just going to ask if you've you know, any intravascular applications of this. I mean, so you've got you're looking at hollow structures like the bronchus, and let's say the esophagus. And, you know, have you looked intravascular, like aorta, done any work there?
So we haven't put it up a guide wire yet or anything of that sort. We think it's consistent with that because we can see the lifetime even in perfused tissue. So we're not even in highly vascularized tissue we can see signal. You know, in the third hour of my talk, I could tell you about our imaging of the beating heart and watching blood cells inside the heart. So we're trying to exploit what other people would consider autofluorescence or absorption by the blood, test that signal. And so if we can exploit anything about it, you know, using these tools, we can see it. If the optical path is too challenging, then we might have to think if it was on a guide wire or something, you know, having to do a flush of saline or something of that sort to get rid of that signal. What?
Or balloon.
Yeah, or balloon, right.
So for years in coronary arteries, we've been chasing the concept of the vulnerable plaque. I mean, thermal maps fail. We use intravascular ultrasound. Now we have intravascular OCT.
Yeah.
And that improved the resolution, but still, we don't have a good answer yet. So we have virtual histology.
So one of the things that we've been doing is this technique that, well, so there's a beating heart. Let's see. Is that true? Yeah, so this is an embryonic heart developing, but so we've been building a lot of tools to try to image the heart as it patterns and develops, but the main thing is to use phase variance OCT. Let's see if I can get past all this. So instead of using intensity-based OCT, this looks at the phase of the signal.
So that's a retina?
That's a retina. That's a human retina. In fact, let's see. Since the vessels are different enough, that's not me. That's one of my ex-postdocs, I think. But anyway, this is just looking at the variance in the phase of the signal that comes back. Intensity-wise, you wouldn't see it. And what we would love to do on the vulnerable plaques is to look below the surface at the mobility of the scatterers. And what we've been seeing in inflamed tissue versus cells that are starting to execute the cell death cascade and a number of other settings is at least there's a surrogate marker in the mobility of the scatters. And I've been very interested in the vulnerable plaque question if we could see structural features that normal OCT or ultrasound are a little bit blind to. It may be that ultrasound's the more informative in some ways, and it may be that combining the two, and there's active programs at USC to try to combine the two, and also at Irvine now that I think about it, that really might be able to jointly interrogate. But I would argue that if we went into this phase regime, so this is this is 20 seconds of, oh, well, the pixels are getting all screwed up. But that's 20 seconds of imaging through a non-dilated pupil. So what we've been trying to do is make something that could screen quickly. Since it works quickly, it's something that we think could be used to interrogate plaque. But all we needed now is an instrument that's phase stable enough that we could do the algorithm.
I mean, the big question, of course, is that MIs, heart attacks, occurring lesions that are 40, 50%, not the 70, 80%. So
Same with strokes.
Exactly, same with the stroke with the carotid. So if you find a way to just detect that vulnerable plaque, that you may be able to stent it or operate or whatever.
Right.
I think it was Fujimoto's lab or Boma's lab, no, Boma's lab years ago that accidentally has an OCT series of a plaque rupture. I hope not caused by the OCT, but anyway, so I mean, I think it could be a really nice way to go after it, but we haven't been active in that.
It's interesting because I don't know, I know that USC has a lab that is dedicated to just creating intravascular ultrasound imaging catheters.
Yeah.
I mean, and we never interact with them. But you see
Well, I mean,
exactly.
Is Jesse involved in that, Suku?
I don't think so.
But
So
so
the
there's a
components
lab,
are-
they're working together with one of the labs at
In
Irvine to add the
yeah,
OCT to it. So it would be great to, getting him to come and tell you about what
Well,
he's been
they
doing.
had
would
some
be
of
great.
their students and engineers come to the lab with us to look at OCT and IVUS images.
Yeah.
This
The
is
other one that could be really fun, although right now Odolo has taken him hostage, is there's a person, Brian Applegate, who's a real innovator in OCT technology that's making intraoperative OCT for the ear. And so he's made very small, very stable, and they can go in and image the vestibular organs and the cochlea with that. And he's just upped the game in the imaging, Applegate, and so he could really be somebody to bring in and steal things from, if you can get him away from Ogloi. But anyway.
I mean, this is just, this is neo kind of living pathology, and I mean, it's really going to do away with sort of in a lot of ways with pathology lab, as you get this sort of active live imaging, but completely coincidental to our initial visit. That Karen is here, and she's been running an NIH-sponsored project that we have. We're almost done and rolling already, where we're using an Irvine
using the modulated imaging,
hyperspectral device. This came. We also were doing work in Arizona on this with Craig Weinkauf, one of our old residents as well, and that device appears to be possibly even more sensitive than things like that we have right now, which are imperfect, like some hemodynamic measures, like toe pressures, but also even TCPO2, transcutaneous oximetry, and skin perfusion pressure. So that's instant now, and it's giving us sort of the oxygen, it's almost the oxygen anatomy of tissue immediately.
We've used this instrument to look subdermal at the vasculature, and so it works nicely too.
And what kind of depth? What kind of depth can you get for?
So we haven't established how deep it could go depending on the wavelength. You should be able to go millimeters. Probably 4 millimeters would be a reasonable limit and still get good performance. We've been trying to get this through the approval hassle, but we had people come by at one point from a government agency and say that they were having a problem with some things of fingerprint readers because dead fingers still have fingerprints.
Right.
And so we were making for them read the vasculature
for a
subsurface.
biometric, yeah.
That's a biometric
Well,
for a
but you know,
while.
so Andreas Heisler, those guys in Colombia,
Mhm.
we've been looking into what's called VOTIS, which is like a tomographic hyperspectral. Are you aware of that and kind of the science behind it? Because the data from that really look compelling because it's literally like a CT of instance, almost a CT of anatomy of a three-dimensional structure that you can. I mean, it's big, you know, like a leg.
Yeah, no,
or something,
there's,
yeah.
I think there's a lot of techniques, and it'd be fun to do a shootout. So modulated imaging, for example, is great at seeing organ scale or appendage scale sort of things because it gets signal out of, that scale. We've been trying to push it down to where we could watch at the cellular or subcellular level, and it'd be really fun to see if there is a middle ground between the two, sort of like the ultrasound OCT game,
Yeah.
where you might be able to combine their approaches for the diffuse regime, where you're getting a average sort of result, and this where you could get the subcellular detail and be able to combine it and try to see what could be done.
Have you looked at any nail beds with?
We haven't, but I bet that would work pretty easily.
Yeah, because that's the way they used to do capillaroscopy, right?
Yeah.
Mm-hmm.
And that's-
Yeah, but it'd be fun to just look at a number of different settings. One of my colleague Dan Swartz up at UCSF has been talking to people about looking at margins of burns. I think the same thing's been looked at UC Irvine to look at the vascular status there, or near reconstructive sites to understand where there's been dieback of the vasculature and really actively,
Yeah.
sense where there's still a
Boy,
good
well-
tissue.
Welcome to our world.
Yeah. Well, oh yeah, I guess that is your world all the time.
Yeah.
That'd be good.
Every day.
Other questions? So if anybody's got problems or time or fellows that would like to work a little bit in this world and a little bit in that world, like next week we're meeting with Amir Goldkorn. We've been able to image in his settings using some of these tricks and seeing the metabolism of the cells, very quickly seeing their response to chemotherapeutic agents, looking at heterogeneity in the circulating tumor cells, questions that are easy for him to ask and almost impossible for us to ask, and so we're always looking for partners of that sort. So if anybody has free time, or has time that you're willing to make a little bit free, we'd love to host or figure out a way to bring toys up here to try things up here.
Yeah. Where's your lab? Your lab is on the University Park campus?
Yes,
No.
so I'm one of the founding members of in the Michelson building. So
okay.
we just moved in and
Yeah.
we're trying to get going there.
Yeah.
But I'm here and interacting normally weekly with at least somebody at the Stem Cellers E&I, and then at Children's Hospital most Tuesdays. We've got an imaging center there. And the idea there was to make an imaging center that's staffed and ready to do things, so when people do have a little bit of release time, they can storm in and use a setup that already has expertise on it and the equipment.
Well, I mean, I think we could see about doing something like that.
Yeah, no, that'd
We
be
already
great
have
fun.
gadgetry kind of coalescing up in our, in the SALS clinic now, and I can't see why we can't roll that stuff out now. And you have a lot of other things there on the plastic side too that probably could be really
No, it'd be
synergistic.
really, I think it'd be just dynamite.
Yeah, because
to
that's the
try.
biggest problem, right, is getting,
Right.
is being where the people are, the patients are.
Right,
'Cause that's
well,
where the
yeah,
problems are.
and to have release time and you walk into an empty room, you can't do a whole lot of science, and we need access to the people, we also, to the subjects, we also need, good insight into what are the problems we're solving. If I just ask the engineers to solve something, they will come up with a solution to a problem you don't have.
Yeah, it's the old joke is that engineers that aren't around patients solve yesterday's problems tomorrow. So,
Yeah, okay, that's a good way of putting it.
But maybe you can figure out a way for Karen not to be enrolling so many people that she keeps breaking the modulated imaging camera. 'Cause that's what's happening now. We're seeing so many. We're
Well,
enrolling so
there
many people.
could be several different causative things there, so we could do the experiment.
It's a good problem to have.
Yeah.
But Professor Fraser, on behalf of all of us, this was mind-blowing. I look forward to further collaboration.
Thank you.